All right, so a couple of things before we get started. I don't know if you saw the announcement last night or not, but the exam scores are up on Blackboard. Now, I got an email like really late last night saying that for some reason the take-home score may not be showing up. I don't know the reason for that. Everything on my end seems to suggest that you guys should be able to see it. Are people able to see it? Okay. Maybe it was just something unique to that one person. But if anything comes up, let me know. I do know, though, at least the total score for it, or it is a part of that total score. So as of like right now, that total score is a good reflection of is a good reflection of the midterm score that I'll be putting into.
Attention, this is the test, the outside summary or TA system. This is only a test. Thank you.
So it will be a good reflection of, there should be no surprises, I guess. I'll be plugging those in later today. Just so you know where I'm at with things right now, I got through like 8 of the videos last week on Sunday and then dropped them completely to start working on the exam, so I'll begin to work on those again this week and try to be going through a few each day. So my hope is that by next week I'll have those done. It just depends on other life situations. Okay. So other than that, I'll talk a little bit about the exams on Monday once I hand them back. And if you have any questions other than that, let me know. For homework for Monday, it's a blog. I posted the instructions this morning. I realized I was a little behind on getting things posted for assignments, so it's now posted through Wednesday's lecture, and then I'll make sure that over the weekend I get more posted as well. Through next week, Wednesday, so meaning homework for this weekend, and then Monday, Tuesday, it has been posted, so you can go ahead and get started with them if you would like to. The one for Monday's lecture is another 2-parter. So the first part's due Saturday at midnight. The next part is due Sunday at midnight. The Sunday part is just a response to a partner's comments. Okay, and this is all it's a blog version this time. Any questions about what's coming up? Okay. So you may or may not have noticed there's somebody that's been coming into class that like just sits in the back of the room and has been kind of unannounced so far. So Kathleen in the back there is a graduate student at UW right now. She's going to be doing actually a couple of lectures down the road. So she's just been kind of watching to see how the class runs and how I interact with you or don't interact with you. So. So she's going to be doing one of the lectures on viruses down the road. Her expertise is actually in human papillomavirus, so she'll be doing a lecture on some of the human viruses. And then she'll come back toward the end of the semester and do another one on vaccinations when we get to that point as well. So I just wanted you guys to know who that is, so you don't have to like shun her by not knowing who she is. Anyway. All right, let's get started. So today we're going to talk about gene expression. We've got one objective. Describe how bacteria regulate the expression of genes. And we're largely going to do this through the context of the lac opera. I took a look at the homework. Some looks good. There are some corrections we need to make. So what I wanted to start with is, let's go through this chart and let's actually talk about the 4 different conditions that we usually discuss when we talk about the lac opera. once we do that, I want you guys actually to take that list of trends that you created, and I want you to make revisions if revisions need to be made. You may notice on the wikis, if you've paid attention to them in the past 10 minutes, I've gone back and at least finished filling in for some groups where there were, like, this isn't so much a problem in this class, but in groups of 3, or if somebody forgot to do the assignment, I filled in one of the rows for the group. And then if there was anything that I had different results for than what you had, I made corrections to that already. So that should be updated. If you see nothing, it's good as stands right now, okay? So let's just start with the basic premise here. What is the lac operon? Or what is an operon in general? I kind of briefly mentioned this term on Wednesday. It's a thing.
Sequence of genes.
Sequence of genes. Usually one promoter, numerous genes downstream. So they're all regulated by one single promoter. What binds to a promoter? Just to review something again from Wednesday. Yeah, it's the RNA polymerase, and specifically the sigma factor brings the RNA polymerase to that promoter. We talked about, for example, the -35 and the -10 regions of the promoter being important for the RNA polymerase to bind. Okay? The lac operon was one of these first operon systems to be discovered, and so this is the one that we know probably more about than any other operon system. There are nuanced differences to them, but they all kind of function in the same way. You can think of them as like toggle switches. We're going to talk about it simply as on or off. One thing that we won't talk so much about, but is true, there's a lot of shade of gray here, okay? I had you answer like yes or no, the genes are going to be expressed. It depends on the levels of the different metabolites. So for example, glucose in most organisms is preferred as a carbon source over most other monosaccharides. Lactose, on the other hand, will be used when glucose is gone. Okay? So glucose you can kind of think of here as a major, or kind of, somebody actually in one of theirs described it as the alpha dog, that it's like the major regulator. In some ways that's kind of true. We're going to talk about this and look at how really this operator system, or this operon, is really a toggle of 2 switches. 2 different switches that control gene expression. So what we're going to do is we're going to start by just kind of drawing out the basic features of this system, and then we'll begin to talk about what happens when glucose is present or absent, what happens when lactose is present or absent. Okay? So the operon itself has I'm going to just draw an arrow here as that transcriptional start site that we talked about last time. 3 genes downstream, LAC-Z, which is beta-galactosidase, Y, and A. And then it's got a promoter region, and it's got what we call an operator region, or LAC-O. So LAC-O, promoter. And we already said that one of the proteins that binds to this at the promoter region is that RNA polymerase. What else can bind to this operon system? What other proteins do we find? Devin? Yeah, CAMP and CAP. So CAMP is the regulator, CAP is the protein. So CAP, and CAP stands for catabolite activating protein. There's also, sometimes in the literature you may see another word that, or another abbreviation that's used, which is CRP. So that will bind to CAMP, which is cyclic AMP. And then what else binds here? Yeah, the LAC-I protein. So LAC-I you can kind of think of as like a doorstop, or just a stop in this place. When it's present, its job is serving as a roadblock to prevent RNA polymerase from moving. And this is the molecule that lactose will bind to. So we've really got here two binary switches. One is LAC-I, the other one is CAP. One activates transcription, the other one actually is involved in repressing transcription. Which one represses transcription when it's bound to this promoter? LAC-I does. When it's bound, it prevents the RNA polymerase from moving. Okay? CAP, on the other hand, is an activator. It recruits, or it helps to bring the RNA polymerase to the promoter site. In other words, it's kind of almost like, I describe it in some ways like a molecular beacon. It's telling the RNA polymerase, come here, come bind, so that we can transcribe these genes. Okay? Any questions with this basic concept? These are the major role players in this process. Devin?
Did you say that was CAP or CAMP? That was like one RNA?
CAP, the combination of the two of them, I guess I should say. Okay, so what I'm going to do is I'm just going to move ahead again and just draw draw another one of these. And we're going to just focus first on lactose and the LAC-I. We've already said that LAC-I is kind of like a roadblock. It binds to the DNA to stop transcription of these particular genes. When does it bind? So in what cases does LAC-I bind to the promoter? Yeah, when lactose is absent. So lactose we could describe here as an inducer. It's going to turn on expression, okay? And that's one of the terms, I put that as a list of, in the list of terms for you guys to look at. So inducer, lactose is an inducer because it's turning on the expression of these genes. And this should make some sense in some ways. And I guess to understand the context of it, the purpose of those LAC-Y, LAC-Z, and LAC-A genes are to break down lactose into two monosaccharides, glucose and galactose. Both of those can then be used as carbon sources within the cell to produce ATP. So the only time that the cell would want, quote unquote, to actually expend energy in creating these genes would be in cases where lactose is actually present, where it's going to be beneficial to produce these genes. Otherwise, why waste the energy in doing so? Okay? So the only time when we get activation then is when lactose has bound to LAC-I. And lactose in some ways here is like an allosteric molecule. Anybody remember what an allosteric inhibitor or an allosteric activator does? Where does it bind? The other term we used was non-competitive. Yeah, anywhere but the active site. And we said, what does an allosteric active inhibitor do to the active site? Good. It changes its shape so that it no longer can function, right? Well, we describe lactose as an allosteric molecule. Allosteric molecules in general are any molecule that changes the shape of a protein and in turn turns the protein on or turns it off. In this case, when lactose binds, it changes the shape of the LAC-I protein so that it no longer can bind to the DNA. So it takes the amino acids that LAC-I is using to bind to that DNA, and it basically kind of like folds the protein in a way so that those amino acids are no longer available to bind to the DNA. So it changes the shape of the protein. At that point, transcription could occur. Okay? I'll say could instead of does occur because there's a caveat here. So this one is just a simple binary switch. Lactose present, LAC-I comes off. Lactose absent, LAC-I binds to the DNA, inhibits transcription. Okay? Any questions on that switch? Okay. Let's go to the next one real quick. So our second switch here was cap. And cap is bound to the DNA when it's bound to cyclic AMP. When does cyclic AMP get produced within a cell? Yeah, when the energy state is low. So cyclic AMP is just a part of a continuum of basically energy concentrations. Energy inside the cell, typically we measure as what? What is the main source of energy? ATP. And it's the phosphates on the ATP that provide the energy for reactions to take place. When a phosphate comes off of ATP, it forms ADP. When the next phosphate comes off to provide more energy, the molecule goes to AMP, and then we get this little circularization reaction that creates cyclic AMP. So cyclic AMP is typically a kind of, we call it an alarm mode sometimes. Like hormones change function of cells, it's an alarm mode to tell the cell that energy levels are low. When that happens, the cell has to somehow produce energy. And so in this case here, when cyclic AMP binds to cap, this is going to take cap and put it onto that promoter region. Cap has some amino acids that allow it to interact or to bind to the RNA polymerase. And so in doing so, this is how we get that recruitment of the RNA polymerase to this particular site for transcription to occur. When energy levels are high in the cell, so when there's a lot of ATP, there's not a lot of cyclic AMP, and therefore cap becomes unbound to the DNA. RNA polymerase isn't actively recruited. Okay. So putting this all together then, what does this mean in terms of that table? All right, so here's that table that we looked at. Let's start out just looking at glucose and looking at cyclic AMP and cap. Okay, I like to break this down usually by the carbon source first, and then go instead of looking across. Even though I had you guys look across in the rows, but if glucose is present, is cyclic AMP present? No, glucose is going to produce a lot of ATP, and so in that case, cyclic AMP levels would be low. So we can say no and no here. Would cap bind to the promoter then? No in either of those cases. But when glucose is absent, that means that the cell is nutrient deprived. Probably it's burned up its allowable amount of ATP, and now we would expect cyclic AMP to be present and cap to be bound to the DNA as well. If cap is bound, is RNA polymerase being recruited? Yeah. If cap is bound to the DNA and is bound to cyclic AMP, we'd expect RNA polymerase to be recruited as... Oh, I have this backwards. Do I? No, I'm okay. I'm just going to check this real quick. I feel like I've made a boo-boo, but I don't know. No, I have, and that's okay. We're going to go with it because you're going to correct me. Okay. What about, in what cases is lac-I bound? So lac-I is bound when lactose is absent, yes. So when lactose is absent, we get binding, yes, yes, no, no. And then we would take a look at the expression. I've made a mistake with the RNA polymerases. Which of those is incorrect, given everything that we've talked about so far?
Yeah, when glucose and lactose are both absent. Why, Austin?
Because if you treat the chronic, this isn't there, so it can't be promoted.
So the RNA polymerase, yeah, what really happens at this point is there's a competition for binding between the RNA polymerase and the LAC-I protein. Because you've pointed out that in the absence of lactose, LAC-I will be bound to the DNA, and it actually is competing. There's actually a little overlap in the binding sites between the RNA polymerase and LAC-I. And so in this bottom case here, in the absence of lactose, what we would actually find is that LAC-I binds to that promoter, outcompetes the RNA polymerase, and so we get no binding of the RNA polymerase in this case here. Okay. Everybody okay with this so far? Okay. And then does LAC-X, LAC-Y, and LAC-A get expressed? In which cases do we find expression? When lactose is present, okay, and is glucose present or absent? Yeah, when glucose is absent. So, in other words, the only time when we find expression is actually when the RNA polymerase is not only bound but being recruited by CAP to that promoter region. Now, I want to point out one thing here. Some folks had put in this top row that, well, yes, if lactose is present, yeah, the RNA polymerase can bind, and yes, we'll get some expression. Okay? This is one of those shades of gray. So let me show you real quick. What I'm going to do is I'm going to draw out some results here to show you what actually happens. We can measure this, whether this promoter is active or not. And the way that we do so is by measuring how much LAC-Z is produced. And LAC-Z produces beta-galactosidase, which is an enzyme. I'm just going to keep that table right now and draw this. Oop. All right, so here's a little chart. No, that's an awful picture. Oh gosh, now it's even worse. We'll just erase the whole thing. Okay, start over. So when glucose is present or absent, and when lactose is present or absent, here's what we actually find. This is the amount of protein that gets produced, or relative amount. This is typically what we find. Now, and again, this is just kind of relative. We usually find that, as we've already stated, in the absence of glucose and the presence of lactose, we get really strong expression of these genes. Those are prime conditions in which LAC-I is removed, CAP has been recruited to the promoter, and we have, at the same time, RNA polymerase then being recruited by CAP to that promoter region. When lactose is present, typically dead as a doornail. You can't typically measure any activity at all. But when glucose is present and lactose is present, sometimes we get a little bit of what we might call leakiness. That there's some activity, but just not really a lot. And this can depend on the strain of E. coli that you're working with. But the reason for this is that the RNA polymerase still has some ability to bind to that promoter, even when CAP isn't around. CAP just serves as an extra recruiter to increase the likelihood that RNA polymerase is going to be bound to that promoter region. And so that's why we typically see results something like this. Now in a binary world, if you want to just know, is the answer yes or no? The only time we see expression in a binary world is in cases where glucose is absent, but lactose is present. Okay? And if that's how you want to memorize this, I will not fault you for doing it. I won't take points off on the exam for it. Because I know that's what really matters is the exam, right? Not the lactose, not this amazing, amazing system. Any questions with this? Kaylin? Yeah, sorry, this would be LAC-Z expression or LAC-Z activity. Thank you. I expect you guys to label those axes. I should probably hold true to my own rules. Any other questions? All right, let's take a look at what if. What if this gets screwed up? Good, better, best question. So, imagine, so how might the regulation of the LAC operon change if a point mutation, in other words, a DNA mutation occurred that affects the LAC-I protein, and specifically, it blocks LAC-I from ever being able to be bound by lactose. Okay? So what I want you to do is go ahead and start out with what you know. Think about the conditions of what you do know about this. And then I'd like you to go ahead and state what's going to happen when this gets screwed up. And I'll give you a few minutes to work on this, and you can feel free to work with somebody else as well. And then we'll go ahead and talk this one out. Okay? All right, go ahead. Sorry.
That guy would always be bound.
Yeah.
Does the last run have to be
Okay. So let's go over, let's just put together a list of some basic trends then that we observe in this particular model. So, lay them out. You weren't all talking about nursing interviews again, were you? Kaylin, go ahead.
So when glucose is present, the camp and the cap do not bind to the promoter.
Okay, good. So when glucose is present, cap and camp don't bind. And you could say vice versa, when glucose is absent, these do bind. Absolutely. What else? Right. Yeah. We are saying that when glucose and lactose are present, that yes, no is the correct answer there. You can say no, you can say very little will bind, but if you want to think of this in binary terms, no is the best answer. That's how I phrase it, because I'm a stickler for detail, I suppose. Go ahead, Meg.
So could one of them be the RNA really bind when just lactose is present?
Yeah, yeah. So RNA polymerase binds only when lactose, is present. Ashley, do you have another one?
I was going to say, lac I belongs to the operator.
Okay, good. So lac I binds. And the abbreviation for that operator is actually LAC-O, so I'll write that this time. Lac I binds the operator when lactose is absent. Others? Meg? Yeah, and another way we can state that too is the only time those are present is when the polymerase binds to the promoter as well. Yeah, so lac Z, Y, and A only transcribed. Oops, I don't know how to spell. No! Such a damn travesty. All right, like ZYA transcribed only. Okay. Any other ones? There are many you can draw from this, and some of them are overlapping. Just like we pointed out with this last one, you could say that it's dependent on just the lactose, or you could say, well, it's the RNA polymerase. And it's only when the RNA polymerase actually binds. So keeping these trends in mind, I think, is one way to kind of mentally compartmentalize what's actually going on here. That the practice problem that we went through, that actually was an exam question from last year in the fall, I think. We did not cover this in the spring. I tried something else, and it just failed miserably, so we're not going to do that again. But operon's one major way in which regulation occurs. I want to point out here, though, that genes are regulated in multiple different ways within prokaryotes. the operon system is one. There are other examples. I want to just show you one other one. So bear with me for 2 minutes here. We talked about on Wednesday how translation of an mRNA starts from what? Where does translation begin? You may know this from the past as well. What's the start code on? AUG. It starts from an AUG. Great answer. Okay. The ribosome has to actually be able to bind and read that AUG. Some genes in bacteria, like the gene that encodes for the OMP-F or the porin protein known as OMP-F in E. coli, where that AUG is, some cells will encode what we call small RNAs or antisense RNAs. And basically what those small RNAs do is that they bind complementary to an mRNA. So if the OMP-F here is in the 5' to 3' direction, this one, UAC, nope, that's not right. That's 5' there. That's 3'. Would be antiparallel and complementary to this. What does this small RNA then do to translation? And the answer is it blocks it. By creating this base pair here, the ribosome cannot see where the start codon is. So basically masks or inhibits the start codon from even being read by the ribosome. I point this one out in part because there's just a number of different mechanisms that can be used to regulate genes. It's not only at the level of transcription. It can occur after transcription has taken place, like in this example here. Claire?
Did you give a name for what this is called?
These RNAs are small RNAs. That's kind of, actually that's the term that gets used for bacterial ones. This one's called McF, not that it matters. It's just a really cool gene. That's the only function of this RNA. It encodes no protein whatsoever. Its only role is to inhibit the expression of OMP-F. It's pretty cool. At least to me. All right. And to my 6-year-old. So, with that said, major points today. Lack operon. Okay? I think that list that we just put together, that list of trends, is a really good summary of what we've talked about. If you're looking at how to study this one, take a look at the review questions. And also I have my good, better, and best responses to that question that we just did as well about the mutation. You can take a look at that to kind of gauge where you're at with the information as well. On Monday, we will actually spend time talking now about mutations and the effect of mutations on protein structure and function. The homework assignment is one where you're assuming that there is a gene that, or there's an amino acid that's being mutated in the active site of an enzyme. You have to describe what effect does that have. So how does it change the structure? In other words, how does it change the amino acid? And then how does that change the function of the enzyme as well? Some of them you may find, and we'll come back to this a little bit later, but I want to also point out one last thing as you're looking at these. Amino acids, we can break them down into ones that are hydrophobic, hydrophilic, acids, or basic. And that's based on the side chain. So you may want to pull up a chart of the amino acids, and if you see a change from like a valine to a leucine, they're both hydrophobic. And so you might hypothesize that, well, there's a change in the amino acid. It's not going to be much, though, because the properties of these amino acids are similar. Okay? That's what we're going to spend some time on Monday and talk about this again in the context of antibiotic resistance. All right. Have a great weekend. Enjoy the weather. I'll see you guys on Monday.